Today we'll be detailing the importance and process of the pentose phosphate pathway. The process occurs in all cell types and tissues. It is part of the carbohydrate metabolism, running parallel to glycolysis, and is even responsible for 30% of glucose metabolized in the liver. The process is unique due to its lack of production or consumption of adenosine triphosphate, or ATP. The pentose phosphate pathway is instead responsible for producing ribonucleotides and NADPH. The ribonucleotides are precursors for DNA and RNA. The NADPH is a reducing agent needed for fatty acid synthesis and acting as a coenzyme by supplying electrons. The process occurs in the cytosol of cells and begins in the first of its two phases, the oxidative phase. First, an oxidized glucose molecule is shunted from glycolysis. The glucose 6-phosphate molecule is oxidized using NADP+ in a reaction catalyzed by the enzyme glucose 6-phosphate dehydrogenase. This produces 6-phosphogluconolactone and a NADPH. Six-phosphogluconolactone undergoes a condensation reaction catalyzed by gluconolactonase, used along with a water molecule that forms a H+ ion and a phosphogluconate. Phosphogluconate is then oxidized by NADP+ molecule, and the reaction is catalyzed by 6-phosphogluconate dehydrogenase. The reaction forms a NADPH, a carbon dioxide molecule, and a ribulose 5-phosphate. The pathway now enters its next phase, the non-oxidative phase. The ribulose 5-phosphate molecule is converted into either ribose 5-phosphate or xylulose 5-phosphate. Ribose 5-phosphate is a main product of the reaction necessary for producing DNA and RNA. Each of the molecules can be converted into sugars in enzymatic reactions so that they can supply glycolysis or act as precursors for amino acids. Further conversions of sugars can occur to provide more to glycolysis.